Hello students, let us continue. Chapter 2, Is Matter Around Us Pure? So today we are going to discuss our next topic of separation, that is chromatography, including that distillation, fractional distillation, etc. So let us start the topic. The very first topic is the dye in black ink a single color or not? So to discuss this, let us have an activity 2.7. Take a thin strip of filter paper. Draw a line on it using a pencil approximately 3 cm above the lower edge. Put a small drop of ink at the center of the line and let it dry. Lower the filter paper into a jar or glass or beaker, or we can say test tube also, containing water, so that the drop of ink on the paper is just above the water level, and leave it undisturbed. Watch carefully as the water rises up on the filter paper and record your observations. So on observing, the water rises on the filter paper, the particles of ink also rises, and different colors are obtained on the filter paper. Why? Because ink contains dye, that is the mixture of two or more colors. And the rise of the colored spot on the paper strip shows that the colored components of ink are soluble in water. So hence, we can say that the ink is made up of different dye colors. The more soluble part rises faster and gets separated from the mixture. So, chromatography is the technique used for the separation of those solutes that dissolve in the same solvent. And some applications of chromatography to separate colors in dye, pigments from natural colors, drugs from blood. Next, the separation of mixture of two miscible liquids. Miscible liquids, that means two or more than two liquids dissolve in each other in such a way that we cannot identify them with our naked eyes. So to discuss this, let us have an activity. Activity 2.8. Let us try to separate acetone and water from their mixture. Take the mixture in a distillation flask. Fit it with a thermometer. Arrange the apparatus as shown in the figure. Now heat the mixture slowly, keeping a close watch at the thermometer. The acetone here vaporizes, condenses in the condenser and can be collected from the condenser outlet. But water is left behind in the distillation flask. So what we have observed here initially, the mixture appears boiling and the temperature rises at initial stage on heating the apparatus. Then the thermometer reading becomes constant at about 56 degrees centigrade. Why it is fixed at 56 degrees centigrade? It is the boiling point of acetone. Here one question arises, why do the two components separate? Actually, the two components, for example acetone and water here, separate due to the difference in their boiling points. We know that the boiling point of water is 100 degrees centigrade, while the boiling point of acetone is 56 degrees centigrade. We have just discussed. So, due to the difference in their boiling points, we can separate such kind of miscible liquids with the help of distillation method. Now, fractional distillation, it is the process used to separate two or more miscible liquids for which the difference in boiling points is less than 25 Kelvin. A simple fractionating column is a tube packed with a glass beads. The beads provide the surface for the vapors to cool and condense repeatedly, as shown in the figure. So fractional distillation is the process of separating the components of a mixture into a series of fractions of different volatilities by means of distillation. Fractional distillation of petroleum is one of the example. Next, how can we obtain different gases from air? Here, air is a homogeneous mixture and can be separated into its components by fractional distillation. The flow diagram of this process is given. Then, if we want oxygen gas from air, we have to separate out all the other gases present in the air. The air is compressed by increasing the pressure and is then cooled by decreasing the temperature to get liquid air. This liquid air is allowed to warm up slowly in a fractional distillation column, where gases get separated at different heights depending upon their boiling points. So here the components will separate out according to their boiling points. Nitrogen with lowest boiling point will separate out first, then argon and the last oxygen. Arrange the gases present in the air in increasing order of their boiling points, like so nitrogen minus 196 degrees centigrade is less than argon minus 186 degrees centigrade is less than oxygen, that is minus 183 degrees centigrade, and the oxygen gas gets converted into liquid first as the air is cool. So students, I hope you understood the topic of separation like chromatography and distillation. Thank you for listening this video.